Photosynthesis is a fundamental biological process that enables plants and certain bacteria to convert light energy into chemical energy. It serves as the primary source of atmospheric oxygen and is responsible for the carbon fixation that supports all life on Earth. Understanding the intricacies of photosynthesis is crucial for comprehending plant biology, agriculture, and even renewable energy technologies. In this script, we will delve into the fascinating world of photosynthesis, covering topics such as the role of pigments in light absorption, molecular organization of photosystems, the Calvin cycle, and much more. Pigments play a pivotal role in photosynthesis by absorbing light energy. The most important pigment is chlorophyll, which is responsible for capturing photons from sunlight. The absorption spectra of chlorophyll molecules correspond to specific wavelengths of light, mainly in the red and blue regions of the electromagnetic spectrum. Additionally, accessory pigments such as carotenoids and phycobilins broaden the light absorption range and protect against excess light energy. Photosystems are multi-subunit protein complexes embedded in thylakoid membranes. Photosystem I, PSI, and Photosystem II, PSII, work in tandem to harvest light energy and convert it into chemical energy. Within these photosystems, light harvesting complexes, LHCs, surround the reaction centers, capturing photons and transferring the excitation energy to the reaction center chlorophylls. The intricate arrangement and organization of these complexes ensure efficient light capture and energy transfer. During photosynthesis, the excitation energy harvested by the pigments in the photosystems is transferred to specialized chlorophyll molecules within the reaction centers. These chlorophyll molecules undergo photochemical reactions, leading to the generation of high-energy electrons. These electrons are then shuttled through electron transport chains, which drive ATP synthesis through chemiosmosis and reduce NAD+ to NADV. This process couples the energy from light absorption to the production of chemical energy. Photosystem I is responsible for the reduction of NAD+ to NADV through cyclic and non-cyclic electron transfer pathways. It consists of a reaction center, composed of chlorophyll molecules, and numerous peripheral antenna complexes for efficient light harvesting. The electron transfer reactions in PSI are coupled to the generation of a proton gradient across the thylakoid membrane, contributing to ATP synthesis. Photosystem II is a key player in the light-dependent reactions of photosynthesis as it initiates the electron flow. It consists of a reaction center, surrounded by numerous antenna complexes. SW absorbs photons and transfers the excitation energy to the reaction center chlorophyll, where water splitting occurs, releasing oxygen, electrons, and protons. The electrons are then transferred to the plastiquinone pool, fueling the electron transport chain. The Calvin cycle, also known as the light-independent reactions or the dark reactions, occurs in the stroma of chloroplasts and utilizes the energy-rich molecules ATP and NADV produced during the light-dependent reactions. This cyclical process fixes carbon dioxide into organic molecules, mainly glucose. Through a series of enzyme-catalyzed reactions, such as carboxylation, reduction, and regeneration, the Calvin cycle plays a crucial role in carbon fixation and the synthesis of complex carbohydrates. Photorespiration is a phenomenon occurring in plants under certain environmental conditions, which leads to a wasteful loss of fixed carbon from the Calvin cycle. It involves the oxygenation of ribulose 1,5-bisphosphate by the enzyme ribisco, resulting in the formation of a 2-carbon compound, phosphoglycolate. The respiration processes associated with photorespiration consume ATP and release carbon dioxide, counteracting the efforts of carbon fixation. Genetic engineering techniques have allowed for the manipulation of plastid genomes, enabling the introduction of desired traits into plants. Plastid transformation offers several advantages over nuclear transformation, such as high transgene copy numbers, maternal inheritance, and reduced gene silencing. This technology has immense potential in the targeted improvement of crop plants, including enhancing photosynthetic efficiency and stress tolerance. Photosynthesis is tightly regulated to optimize energy capture while preventing damage from excess light or environmental stress. Various regulatory mechanisms, such as the light harvesting complex protein phosphorylation, non-photochemical quenching, and redox signaling pathways, modulate the efficiency of energy transfer, electron flow, and carbon assimilation rates. These intricate regulatory networks ensure that photosynthetic organisms can adapt to changing environmental conditions. Photosynthetic organisms exhibit remarkable diversity, with distinct adaptations in response to their respective environments. Comparing photosynthetic systems in plants and algae sheds light on the evolutionary relationships and functional differences between these groups. Differences in pigment composition, light harvesting mechanisms, and carbon fixation pathways highlight the remarkable adaptability of photosynthetic organisms. Exposure to excessive light can be detrimental to photosynthetic organisms, leading to a phenomenon known as photoinhibition. Photoinhibition occurs when the rate of light energy absorption exceeds the capacity for energy utilization and protective mechanisms. This imbalance results in the generation of reactive oxygen species, which inflict damage to the photosystems and other cellular components. Understanding photoinhibition is essential for developing strategies to enhance crop productivity and stress tolerance. Advancements in imaging and molecular biology techniques have revolutionized the study of photosynthesis. Techniques such as confocal microscopy, fluorescence resonance energy transfer, and proteomic analyses allow researchers to investigate the dynamics of photosynthetic processes at the cellular and subcellular levels. These cutting-edge techniques provide deep insights into the molecular mechanisms underlying photosynthesis. Photosynthesis is a complex and highly regulated process that sustains life on our planet. From the role of pigments in light absorption to the advanced techniques used to study photosynthesis, our understanding of this process continues to expand. By unraveling the intricacies of photosynthesis, we can pave the way for advancements in plant biology, crop improvement, and the development of sustainable energy solutions.